Okay, so good morning to everybody, and today is a great pleasure to introduce the today speaker, Alessandro Luciani. Just a few words on about Alessandro. She's of course Italian, but I can say that he's a very international guy, yeah, because he was born just in Italy, but then he traveled around the world and he worked around the world. He has been in different institutions. Probably he started at Tigem with the Andrea Ballagio, but then he went to Milano, so went to Raffaele, and then in other institution, and finally he approached in Zurich where he's working. And as the Zurich is working is first for as a fellow, and but from now on, I think he is an independent investigator because he just told me that actually he won a very big and prestigious Swiss grant. So for the future, for the 4 years now on, he will be almost independent. I must say that he met Francesco Tebricione, and Francesco told me about 1 year ago. I met an extraordinary guy that's very, very independent, very bright idea. And so I say, Francesco, okay, look at it and just to invite him. So we tried to invite him several times, but very, very lucky enough to have him here today. He's interested in rare disease, not specific rare diseases of a morphological phenotype, but rare disease in general. And looking at rare disease means an expert in rare disease about lysosome units, lysosome vesicles, I can say. And so this seminar will touch upon several issues and several pathways that can be interested for all the groups of the Belgium. And he will tell us, I think, a little history, a little story from what he started, so especially what he can do or he will do in the near future. And I hope that we start to the collaboration, an endless collaboration, and I hope that we have him many times again. So, Alessandro.
Grazie mille, thank you very much, Professor Capasso and Francesco. It's really a great honor, a pleasure to be here. So not just because I'm Italian, but this is my home. I'm coming from Campania as well. So I was just driving this morning and say, well, what I have to tell to this audience, and I was start thinking about, so tell you my story, but then I realized that probably the best would be just to show you the true line that I have developed together with my collaborators in Zurich about what are the possible new mechanism therapeutic paradigms in rare disease. But before we start talking about that, I want to stimulate a bit your brain and just discuss a bit the number. So rare disease are an emerging global public health priority. And this because if we take if we take under consideration the US population, you can see that the rare disease affect more than 30 million in the US. So meaning that 1 American to 10 may develop rare disease. And if we translate these numbers to the world, so we are talking about 400 million people worldwide. So this suggests that if we take as a whole the rare disease community, they have at least about same prevalence of common disease. Of course, so what's the difference between the rare disease and the common disease? That for all these diseases, we know the causes. So typically we have one gene, and this gene is defective. And then we see the phenotype in the patient. And what is really, really crucial is that many of the patients affected by this rare disease are children. So meaning that we have a compassionate mission to achieve. Well, what about therapies? So all of you are aware about the power of gene therapy. Yes, that's fine. But still, we need a bit of time to make so those strategy real safe. So this means that still we don't have any preventive or curative therapies for this rare disease. And this is something really, really important because you can imagine the cost of the healthcare system to have a complete management of all these diseases. I was just, taking up a few numbers, but probably I need to update because the costs are really, really increasing each time. But just to tell you, the US healthcare system so spend more than $130 billion for the management of people affected by rare disease. So all these premises so suggest us that probably we need to identify new transformative medicine that may address the unmet medical needs of all these individuals. But we were trying over the last year to have a strategy to solve the problem. And we realized that probably we need to take a set of disease that affects the proximal tool of the kidney. So give me just a time, I will explain you so what this part of the kidney does in real life. But if we take a set of disease, rare disease, genetic disease that affects the proximal tool as a paradigm, and if we are able to combine with a translational workflow based on preclinical model systems, so cell and organelle-based functional assay, digital tools, and disease-relevant screening technology and human cohort. So what we could do is we can really understand so the fundamental principle that regulate homeostasis. So the rare disease may help us to better understand human biology. And we can even further so try to see whether and how these regulatory circuits may affect so the development of disease and the pathogenesis. And ultimately, so we can use these paradigms to design new drugs, a new therapeutic strategy for all these affected individuals. And of course, our goal is if those strategies are successful, so we can accelerate them. So the translation of therapeutic agents from preclinical systems to proof of concept in humans. So let me introduce the proximal tool. I know that Professor Capaz, Francesco, they know very well this organ. So please correct me if I'm wrong. So what they do, the proximal tubules of the kidney. So typically they are specialized in nutrient reabsorption. So this means that all the proteins, solids that are normally filtered through the glomerulus are uptaken, reabsorbed by the proximal tubule cells. And this process is crucial for the maintenance of the body homeostasis. And I can tell you that I was really super fascinated by these cells because since I'm a cell biologist, I start my science life touching the lysosome. I realized a posteriori that the proximal tubule cells are the best cellular models to study so the role and the biological function of the lysosome. So let me explain you why this is the case. So let me introduce the players that have do the big game. So we have endocytic receptors that are just located at the brush border, apical pole of the proximal tubule cells. And those receptors are essential because they are the guys that are going to uptake, and so the plasma proteins, including album, for instance, or other low molecular weight proteins from the glomerulus. Once bound, so then they start internalize the ligands, and the complex that is going to form between the ligands and the receptors is able to traffic through trafficking pathways that are constituted of cut and coat pits. And they they are going to meet different organ compartments. So the first one is the early endosomes. And this station is crucial because, as you may know, so based on change in the pH, so the ligands start to be dissociated from the receptor. And with one fate that is going to happen, so meaning that the receptor is able to recycle back to the plasma membrane for starting a new cycle of internalization, while the ligands is trafficking through late endosome to reach the lysosome. And as you may know, so the lysosome are our trash bin cans, so they are able to digest macromolecules since they have a huge amount of proteases or digestive enzymes. And the final result is that the pi products that have been digested by lysosome, they can move back to the cytoplasm through lysosomal transporters. And this action is essential because all these metabolites are going or to be stored in a specific compartments or can be reused for a new anabolic reaction or metabolic reaction. Of course, the lysosomes are not only the endpoint of endocytosis, so the pathway that I was showing you before. They are also receiving material from the intracellular side. And I know that all of you are aware about autophagy. So autophagy is an evolutionary conserved pathway that is crucial for the maintenance of homeostasis because thanks to this pathway and organelles like autophagosome, so all the material that needs to be removed, so exhausted organelle, misfolding protein, so they can be engulfed within autophagosome and sent back to the lysosome for the final degradation. And thanks to this pathway, so the cells try to maintain their own integrity and metabolic homeostasis. And I was realizing that probably the connection between lysosome, autophagy, and endocytosis is crucial because, in one hand, so the proximal tool cells are maintaining the uptake of nutrients. In the other hand, so they are also maintaining the quality control of the organ. And if you just figure out that all this transport process need energy, then you can see how much is crucial the lysosome for the fitness of this cell type. I was telling you that so that autophagy is removing damaged mitochondria, and the mitochondria for the proximal tool cells are crucial because they need energy. And energy is important for keeping alive so the transport across the membrane. So as you can see, so the lysosomes are really the big game changer of the cell because they keep alive endocytosis, nutrient, quality control, energy for sustaining the transport. And of course, recently we start learning also that these organs, the degraded organs, they are really crucial also for maintaining metabolic signaling. And all of you know that mTORC1, the master regulator of growth metabolism, is going to assess the nutritional status of the cells just going and making interaction with the lysosome. Of course, the importance of the lysosome is highlighted by rare disease. And typically, any type of perturbation that is going to affect the biological functions of the lysosome has boomerang on the function of the proximal tubule. So this means that defective lysosomal related pathways can cause proximal tubulopathy. So it means that the proximal tubule start doing their own job. And one characteristic hallmark of proximal tubule is that they stop uptaking nutrients, including low molecular weight proteins. And then we have an hallmark of the disease, that is the tubular protein, so appearance of low molecular proteins in the urine. And And our lab was using all these rare diseases to understand what are the biological functions of the lysosome for the proximal tubule cell have in disease. In other words, so we were using one disease in particular, cystinosis, to understand how the lysosome-related pathways may drive the proximal tubule. So let me introduce you the cystinosis. So cystinosis is, of course, a rare disease and is caused by loss of function mutation in one gene that is called cystinosi. So you can see here. So cystinosin is a canonical prototype of lysosomal transporter that under normal conditions make possible the efflux of cystine from lysosome back to the cytosol. And in the case of cystinosis, then in patients with cystinosis, so you can imagine that the transport is defective because of loss of function in these genes, and then the cystine start to be trapped in the lysosome, causing what is called storage, lysosomal storage. And the consequence, the downstream cascade effects of this storage is that the lysosomes start changing phase, they start becoming obese, enlarged, they lose the function, and this may ultimately cause different dysfunction that affect different organs like kidney, brain, eyes, liver, and muscle. So we have a clear picture of the phenotypes that we can see in patients, and quite surprisingly, so the prime hallmark that we see in all patients, most, 95% of systematic patients, they have problem with the proximal tubule. They have a proximal tubulopathy that is called renal vancomycin. And I want just to give you a short historical point about renal vancomycin because it was discovered by an Italian-Swiss physician at the University of Zurich, just to show you that the cycle is closing at the end. The patients are also developing other complications. So as I told you before, so it's a multisystemic disease. They have problem with endocrine system like thyroid. They start having problem with the muscle, with the brain. But keep in mind that the first complication that we see in patients is the proximal tubulopathy, that if left untreated may progress towards chronic renal failure. So for that, then it's crucial that we take cystinosis as a paradigm, because if we are so good in understanding the molecular mechanism driving the proximal tubule dysfunction, then we can see what are the pathways that may damage the kidney before having kidney or complete failure. So we are quite lucky because currently we have one treatment. So the standard of care, so patients are under treatment with histamine. So that is a kind of a magic pill. So in a way that may enter the lysosome and make possible the emptying of the storage. So in other words, so what it does, histamine is able to bind cystine and through alternative, still unknown transporter may allow the efflux of cystine outside from the lysosome. And although this capacity, although we are able to empty the lysosome, there are still problems. So due to the chemistry of the histamine. So typically patients are under frequent dosing and low compliance. So there are still side effects. So the drug is poorly tolerated. And probably one big effect is at the end, so even if you are able to empty the storage, so the drug seems not to prevent or cure the proximal tubulopathy, neither for the kidney disease. So for that, we need to identify new drug targets and transformative therapeutics for this type of individuals. And then we start thinking about, so what's the best strategy to identify drug targets and therapeutics? Of course, we cannot take humans, so then we move back to the preclinical models and we start consider different animal models. And I like use this slide just because probably we are not aware about which the best preclinical model that we should use. But I suggest you to read this review because you can really see that if you want to move drugs, so new therapeutics from preclinical systems to human, you need to be aware about the preclinical models to use because probably you need to have one preclinical model that is faithfully recapitulating the disease-relevant change that you can see in humans. And using this premise, so then we start assessing the downstream cascade of the loss of function of cystinosi across different species. And I'm going to show you and share with you the data that we have achieved until now. So of course we start thinking about the mouse. So we had a collaboration with Corinne Antignac and in Paris, and she was generating the mice. And then we start using this knockout mouse and try to see whether this model may recapitulate all the disease phenotypes that we can see in humans. And as you can see here, so the loss of function cystinosi is able to reproduce in storage. So you see here we have an increase in cystine content in the kidney of the knockout mouse compared to the wild type. So the lysosome, I was explaining you the lysosome start to be obese, and we were able to recapitulate so this phenotype, as you can see here. So the lysosome, these purple vesicles marked by this canonical lysosomal marker called LAMP1, they look more enlarged compared to the wild type. So the knockout is wild type, and we quantify this event. The other two phenotypes, so defective endocytosis and proteinuria, yes, we were able to recapitulate so these phenotypes. So as you can see here, so the knockout proximal tuber segment is unable to uptake less, so this low molecular weight protein called lactoglobulin. And then so this was reflecting also an increase in the urine of the level of C16, which is another low molecular weight protein. So it seems that the knockout mouse model is able to show the disease-relevant phenotypes that we can see in humans. Of course, we are mechanistic guys, so we want to see what are the mechanisms behind this phenomenon. And then we start considering the cellular systems. So, and we had the big problem here because of, as you may know, if you want to start working with cellular systems, you have immortalized cell lines. But then, if you consider the job that the proximal T cells do, you can easily realize that for keeping alive the transport, you need to have a super polarized and well-differentiated cell type. So meaning that the cell types have to do endocytosis, endocytic uptake. So we start analyzing different cellular systems. So we have canonical cellular systems that you can find on the market, like OK, HKQ, 2, LCC, PK1, but just names. And once we start comparing these immortalized cells with the primary cell culture, so that we developed in-house, we had a big surprise. So all these cells that have been used for modeling mechanisms, pathogenetic mechanisms, when we start looking at endocytosis or endolysosomal proteins, you can see a huge difference. So our primary cell cultures start expressing better so the endolysosomal endocytic receptors compared to the canonical cellular system. And then we did some functional assay where we start comparing the level of endocytosis and proliferation of our primary cells compared to the canonical immortalized cell lines. And as you can see here, so our cellular system recapitulate perfectly the endocytosis uptake, and they have also low rate of proliferation. So meaning that this type of cellular system can be used as a blueprint to start modeling possible disease mechanisms that may drive, proximal to global. And then we start using our primary cell culture. So we derive those cells from the knockout mouse kidney, and then we start using different cell versus organelle, including lysosome-based function assay. And what we saw is that the loss of cystinosi in the primary cell culture system induce a change in the dynamics of the lysosome. As you can see here, so the lysosome start occupy the perinuclear region of the cells in the knockout conditions, while the wild type condition, they are spread throughout from the knockout cells using immunopurification and lysotag epitope tools. And as you can see, so we have more lysosomes, but those lysosomes seems to be dysfunctional, because if you look at the level of mature cathepsin D, one of the lysosomal enzyme working within the lysosomal lumen, so you have less mature band in knockout compared to wild type. And they start accumulating the autophagosomes or autophagy garbage. I was explaining you the lysosomes are an endpoint for autophagy. So if they are not working, they cannot digest, then you have an accumulation of autophagosomes. And then we start modeling the disease mechanisms that may explain the proximal to the dysfunction. We run some omics analysis. We try to be as much we can unbiased. And what we found from our omics approach is that the loss of cystinosin and the storage of cystine seems to regulate pathways that are involved in cell cycle, proliferation, whilst at the same time seems to downregulate pathways that are crucial for membrane trafficking and transport. So the main activity of the proximal to the cells. And then we run some biochemistry imaging approach to validate those omics data. And as you can see, here we have a huge increase of proliferation and the differentiation of markers. Here we take as a biomarker BMT. And when we look the proximal to the segments, so we have that those cells start to be high proliferative while they start losing the endocytic receptors, including the nella. And then we run other analysis just to validate that there is a reprogramming, cellular reprogramming in the knockout cells where proliferation seems to be highly activated while differentiation seems to be downregulated. Of course, a posteriori, we realized that the mouse model was, yes, good for modeling the proximal tubulopathy, but we realized that the onset of the proximal tubulopathy is not so early, so like the one we can see in patients. They typically, after first year of birth, so the patients with cystinosis, they have proximal tubulopathy renal function symptoms. And then we start realizing that probably if we want to have high predictive preclinical models, probably we need to reinforce our strategy of preclinical models. And then we start figuring out whether it would be possible the modeling, so the cystinosis in the rat model. And you are going to ask why, but so the reason is quite simple. The rats, if you look at the evolutionary scale, are closer to humans, and those are being used, for instance, also for different type of studies, including drug metabolism and physiology. So there is a way that probably the rat may be much more predictive than the mouse model. And then we start moving through the rat model, and collaboration with Francesco Emma, we start to generate the CRISPR-Cas knockout rat model. So essentially what we did is just to introduce a frame shift mutation in the exon 3 of the CTNS rat gene, and as a consequence we just stop codon. So this means loss of the protein translation. So as you can see here, so we validate the genetic strategy by DNA RNA analysis, and we had also functional results. So if we ablate cystinosi, so the cystine should go up. And this was the case because we had accumulation of cystine storage in different organs, including spleen, kidney, and many others. Then I don't have time to show you all the readouts that we are able to monitor in this rat model. I will try just to summarize here. And what I want to share with you is that this really huge message that when we start comparing the mouse knockout for CTNS versus the rat, so we see a huge improvement of the proximal tubule dysfunction. So what in the mouse model was possible to achieve after many months, in the rat we were able to achieve after only 3 months. So meaning that we had really improvement of early onset of proximal tubule dysfunction. And something that we saw also in the rat is that, strangely enough, they have huge cystine storage compared to mouse model, as you can see here. And they had also kidney failure, something that we were unable to see in the mouse model. And we did not see any gender effects related to the phenotype. So this means that in the mouse model, typically the female had the phenotype, while in the knockout rats, so we were able to see the phenotype independent of the gender. So really amazing. And something that we were also a little bit impressed was the life expectancy. So the knockout mouse model, so even if you take longer, so they don't die. While the rat knockout, after 1 year, so they start dying significantly. So then we start considering the rat model as a possible preclinical model to test some drugs. But then we realized that, well, what happens if I have to dose a library of compounds? This would be quite impossible. Imagine the cost that we have to support for testing more than 20 drugs per time in the rat model. So we need to consider probably a more screenable systems if we want to achieve this milestone. And we were considering the zebrafish. Okay, so all the time I start talking about the zebra, so I want just to disclose that zebra is not because I'm a fan of Juventus. In this context, so the zebrafish were super interesting model because I was just looking at how much the biology is conserved across the species, so from zebrafish and human. If you take the disease organ like the kidney, I can tell you that the zebrafish, they have the same repertoire of endocytic receptors, transporters that the human kidney has. So then this is good for us because we are able to model probably the proximal tubulopathy. They have a conserved physiology. They have a conserved drug target metabolism. So meaning that if we have drugs, we can easily test it. And there are other technical reasons that support our choice to implement the zebrafish of a drug screening system. So they are transparent, low cost. Imagine the cost for the rat and the mouse, so huge, amazing. In Switzerland, we pay mostly 25K per month just to maintain the colony, so huge. So the zebrafish are well sequenced. I mean, we know the genome of the zebrafish. We can make multiple transgenic report line. As something that I want to share with you, if used in a certain window of time, so the zebrafish are ethics-free. So meaning that you can test all the drugs that you want without making any deal with your back office or ethics. This is something that we need to consider if we want to accelerate the drug discovery process. Sometimes we are stuck just because we need to deal with the back office and the ethics. And this, I can tell you that Switzerland is really a super strict country in terms of animal duties. And just the last point is that the zebrafish, they recapitulate so the in vivo physiology. Then we consider zebrafish also an in vivo model, means whole organism. So, okay, well, the zebrafish can help us, but how we can now monitor the proximal tubule function of the zebrafish? And then we start here, try to build up so a reporter line that zebrafish. And of course, we take inspiration from the physiology, and what we did is simply we take some low molecular weight protein, we tag with a fluorescence labeling. So in this context is M cherry, and then we package so this expression vector, and then we put back in zebrafish, and we sustain the expression of this protein in the liver. So following the physiological rules that happens in the human kidney. So the low molecular weight protein may be secreted from the liver. So this protein can circulate in the bloodstream, easily filtered through the glomerulus, and then so the protein can be uptaken by the proximal tubule cells to be processed. So we are extremely following the physiological rules, so meaning that we are not creating any artificial or neither we are altering the physiology of the system. And of course, so the easy way that we can assess the proximal tubule is that we can have a double strategy. So in one hand, we can see the fluorescence through a microscopic approach, so tools in the kidney of the zebrafish, and then we can report the fluorescence. So as a readout to see if the proximal tubule is working finally, or easily we can just monitor so the level of the low molecular weight proteins in the zebrafish urine samples. So like something that we do also with humans. So you take the urine, which is the best way to look at what happens in the kidney. Of course, this was a great idea, but we need to validate. And what we did in our lab, so this is just a cartoon to simplify the concept. We use different other, chemical and genetic tricks that we know are able to induce or trigger proximal tubulopathy. So in this context, we use as a genetic paradigm RP2, the megalin. So we remove the megalin, then we should expect not to see the low molecular weight proteins in the proximal tubule. Then it's going to accumulate in the urine. And we use other pharmacological tricks like gentamicin, cisplatin, canonical drugs used in clinics and known to induce renal damage. And all in this context, so genetic versus pharmacological tricks, we saw an increase of low molecular weight protein fluorescence in the urine. So suggesting that we were able to validate so our biosensor as a specific tool able to assess the proximal tubule function of the tubular protein. So it was a good candidate, so to see if we are able now to model so the cystinosis in the zebrafish. And at the same time, so we were making these two zebrafish lines. So we were also modeling the cystinosis in the zebrafish. And just to simplify, I can tell you that if you remove cystinosis in the zebrafish, so we were able to see the storage, as you can see here. And once we cross the proximal tube biosensor with the CTNS knockout zebrafish line, I can tell you that the proximal tubule signs are there, because as you can see here, so we have an increased fluorescence in the proximal tubule nephron of the zebrafish. So meaning that the processing is in such a way somehow modify or alter. And we are also able to see the proximal tubulopathy in the urine, as you can see here. So the zebrafish knockout start having a progressive proximal tubulopathy starting after 10 post-fertilization days. So you can see from 3 months, now we are just 10 post-day fertilization. So we are shortened, so the time we need to model the disease. And of course, we did some genetical tests just to show that this genetic strategy is specific, because if we introduce the wild type, so we are able to reverse the proximal tubulopathy. This I want just to summarize. So what we have found now with these 3 preclinical models, and thanks to the hands of our postdoc, Marine, Patrick, and Zion, so I can tell you that we were able to make a chain of events that are able to trigger proximal tubule dysfunction in cystinosis. So as I told you that, so the loss of cystinosis increased the storage or leads to the storage of cysta in the proximal tubule. I told you that, so as a consequence, we have defective lysosome and problems with autophagy. And then I told you also that we have a shift from differentiation homeostasis to abnormal growth and proliferation. And then finally, we have the proximal tubule leukemia disease development. Of course, we were not happy. We want to see who is driving what. And to try to be as much we can unbiased, then we start using artificial intelligence engine. So we team up with Silico Medicine, an AI-driven company, and what we did is to use these machine learning tools to try to extract as much we can disease biology information and new potential drug targets. So I'm not a big expert. This is another disclosure that I want to give you. I try to understand, explain you in a simple way how these tools work. So so artificial intelligence and machine learning tools, they can, integrate different layers of information. So meaning that we can give omics data, whatever you have. So in this context, we have RNA-seq, proteomics, metabolomics obtained from mouse and cellular systems. And then we have also RNA-seq, for the rat, and we have also RNA-seq for the zebra. So this omics data then can be digested by this tool, and the tool can even integrate this data with other type of layers. For instance, text-based, mining text, KOL, so key opinion leader scores, and all the info related to possible grant, patents, grant funding related to cystinosis. And can digest all this data, and then so the system can even use other score to prioritize disease pathways and drug targets. So for instance, we can ask, okay, please select the druggability score of all these targets. Tell me the targets that can be used for clinical development, tissue specificity, and so on. What it came out from this type of analysis was that on the top of the rank of the top 5 targets, we found mTOR. So mTOR may play a role. And then we decide to look at the direction of the signaling. Okay? And what we found is whatever type of preclinical models that you are going to use, mouse, rat, zebrafish, we have a constitutive activation of mTOR signal. And this was quite cool because actually our findings are going the same direction of other findings reported by other studies, suggesting that imbalance or dysregulation in mTOR activity may drive tubular dysfunction in kidney disease. So suggesting that mTOR pathway, this nutrient sensing pathway, can be really, really crucial for the maintenance of the normal nephron function. So then we start looking how the storage at the cystinosi may regulate mTORC1 activity. And the best strategy was just to get some genetics manipulation. So let me explain you. So we use wild-type cystinosi versus a disease-causing mutant that is called, in this context, G339R. So this mutant, what does to the proximal to the cells? Is able to reach the lysosome. Okay, one is that is unable to mediate the efflux of the cystine from lysosome to cytosol. It's a way just to put their cystinosi and have a cystinoid that is unable to do the job. Then we validate this genetic strategy just by measuring cystine, and I can tell you that under knockout conditions transduced with the wild-type cystinosi, we are able to reduce cystine storage. This is a good sign. Our strategy is perfectly working. We are so good in making the genetic strategy, while the mutant is unable to correct the cystine storage. Okay, the system seems to work. Perfect. We can move forward. But then we start stimulate our cells with nutrients, because you know mTOR is responding to change in nutrients. So if you remove food, so mTOR should be switched off. If you give food, you activate mTOR. And what we can see is that in under wild type conditions, so means the knockout transduced with wild type, so we are able to restore the sensibility of the system to the nutrient withdrawal. So meaning that if you remove food, the knockout cells that have hyperactivation of mTOR, after the reintroduction of wild type, they seems to restore the response to nutrient withdrawal, while the mutant form does not. And then we complete this assay just making immunoprecipitation studies. So we have a target, so wild type, a mutant, and we want to see what are the bits that are going to be bound. And as you can see, after immunoprecipitation, so when we give the wild type back to the knockout system, so canonical proteins called RAGs, regulator components of these two complexes, seems to be less bound to mTOR. So while the mutants still maintain high binding capacity to the components of RAGs and regulator components. And we did the same approach, just taking the wild type and make possible the storage of cystine using modified amino acid, in this context cystine. And I can tell you that we were able to reproduce the same findings that we have in knockout conditions And just I want to highlight this point that even if you take the wild type and give the storage, you can see that in this storage cysteine accumulating condition, you have more binding capacity of the wild type to the mTOR-C1 components. And of course, those components are crucial because, you know, they mediate the translocation of mTOR back to the lysosome for the final activation. Just recapitulating the findings that we found is that the storage of cysteine due to the loss of cystinosis activates constitutively mTOR, and as a consequence, we have an impairment of catabolic autophagy. Then we have accumulation of damaging mitochondrial stress. This leads to disruption of tight junction with final activation of self-fate instructive transcription factor called YPX3 that mediate the shift from from differentiation and homeostasis to proliferation and growth. We had a clear mechanistic cascade of the potential path that may drive proximal to the dysfunction systems. Of course, the approach we did is we want to test our pathway and see if we can rescue a phenotype after drug treatment. And of course, we were lucky because you know rapamycin, so it's one of the best mTOR, like well-known mTOR inhibitors able to modulate the mTOR-C1 pathway. And I want just to tell you that I don't have time to show you all the data, but when we give rapamycin back to the cellular system, so we were able to restore normal level of mTOR. We block all the cascade of events that lead to proximal to dysfunction, and we rescue also differentiation and endocytosis and proximal to the dysfunction. And I want just to show you some critical findings just to convince you about our pathway. We start treating with our rat model with the rapamycin. We did a short term using low doses of rapamycin, something that is crucial because you know there are side effects associated with rapamycin if used at a high concentration. And as you can see in these different panels, so we were able to restore the normal level of mTOR-C1 in knockout treated versus the wild type. We restore the cathepsin D, so meaning that we are able also to restore the activity of the lysosome, and we were able to reverse some differentiation markers and some endocytic receptor like LRP2 in the proximal to segments of the treated rat compared to the untreated one. And just to show you that we had also fantastic effect of the rapamycin on the proximal to dysfunction, we use our zebrafish model of cystinosis, and what we did, of course, we treated zebrafish for short-term, low doses, 200 nanomolar, and we checked for different readouts, and we saw that rapamycin were working finely because we restore, we abrogate the hyperactivation of mTOR, we improve the processing of low molecular weight proteins and reverse the proteinuria in the knockout fish when compared to fish treated with vehicle. So suggesting that targeting the lysosome and mTOR signaling axis may be a tactical pathway in cystinosis. So conclusion, because probably I'm a little bit out of time. So what I've tried to tell you is that the role of lysosome as metabolic sensor that are crucial for the homeostasis of specialized epithelial cells. I have shown you the power of zebrafish rat knockout for cystinosis that recapitulates some phenotypes that are associated with the lysosome, and they may be a powerful tool for accelerating drug discovery development process. I was highlighting the importance to use innovative tools for accelerating drug discovery development, and the artificial intelligence allow us to accelerate the discovery process. And I have shown you also that pharmacological and C1 inhibition rescue lysosomal homeostasis and sign of proximal tubulopathy in preclinical model of cystinosis. And now what we are doing, we are just try to modulate differently mTOR C1 pathway. There are ways able to that can be used for that. For instance, look and think about the time and the dietary restriction, use of caloric restriction mimetics. In that context, we were discussing before with Francesco and also with Professor Capasso about the new use of SGLT2 inhibitors. And we want to use this approach and translate this approach, pharmacological approach, not only for cystinosis, but also for other rare and much more common disease driven by lysosomal dysfunction, including kidney disease. And with that, so I want just to close here my talk, and I want to acknowledge all the people, collaborators, national, international collaborators, funding agency, and thank you for your attention.
I told you that we would hear from our guest today a nice story. Yeah? And it's followed also by several investigators with this one. Because you start with a disease,
yes,
a specific rare disease of unknown origin at that time, often disease not drug,
exactly,
because there were no data about why cystinosis appear in the kidney, but also in other organs. And then you just told us that all the processes were done. You know, the first thing to identify the complex pathway of the system uptake of post-structural. And you start with post-structural, animal model, the artificial intelligence, and so on. So. And now you are open a completely new world, a new trajectory.
Yeah.
In order to save the patient.
Yes, of course.
So this is a very nice story, and it's very common to many of you, and some question about your pathway. I was just curious, because up to now I understood that artificial intelligence may use especially when you want to do a jump from animal studies to humans. Yeah? Now, how you are proposing that artificial intelligence may be used, may be quite useful, also to analyze the data on in-cell culture, animals, zebrafish models. But then how you can jump from this in vitro, in silico, animal materials?
I can tell you that I'm not the big expert in artificial intelligence. I'm like a PhD student now. Try to really spend my time in understanding this type of tools. I can tell you that it's a tremendous revolution, because I was just following a huge AI business meeting in Basel, where, and I took a lot of notes. There are different ways that you can use artificial intelligence. So people now are, at least for certain type of disease, more common disease, they are using prevalently human data sets, so coming from biological fluids of patients. And then what they are doing is just to make replica, so virtual replica of possible disease states in silico. So meaning that you can reproduce human patients. Of course, you could adjust, stratify the patients based on biomarkers or sign of disease. But once you have this stratification, then you can create a digital twin of the patient, and where you can really have a big game, because then you can test pathways. You can of course reconstruct first the pathway, and then you can use drugs and see whether those are able to modulate positively or negatively the pathway. Maybe
No, it's perfect. The way that this kind of things are applied. In your case, I assume that you identify potential target group prioritized target by the emitter actors. So just putting together multiple levels in
yes,
expression, let's say, PPR, protocol interaction. So
yeah, sure.
just to identify what is the goal, where are the goal of the signals that
Yeah.
is measured. So there are different ways to simulation in one case, or this kind of tools, but also integration, data integration, data analysis.
Yeah.
So in that case, it's more data analysis rather than prediction of knowledge, just to homogenize and putting together multiple.
Yeah.
These digital twins, I'm still trying to learn if they are really usable. It's a cool name, and
because still we don't have any paper publication associated.
Right,
This is the point. This is the point. But once I start talking with this guy, they are really super collaborative. So they say, well, we can offer all the technical support. So they are. I can tell you that the computational communities really are open community. I'm so happy to work with them because they want to. Yeah, because they don't have the wet lab. So the unique way to say that, okay, the tool is able to predict is just to go back to the lab, doing experiments, and solidify your idea. But I can tell you also, just to because I was running, I did not have time to explain you also that type of collaboration. Yeah, you are right. So we were in that context, we were prioritizing repurposable drug targets. Because I think that for academic centers like we are, so we are not a company. We are not in a deal to spend millions, if we have, of course, but we don't have. So then the best way to accelerate the process is to have a repurposing approach. And you all know the advantage. But that type of tool, you can make also a first-in-class or novel compounds, small molecules compounds. And actually, this is a part of the grant that I got, where we are now generating new predictive targets, not based on repurpose approach. So just based on targets that we are able to identify once we integrate all the omics. Actually, we are looking for human data set because if I had to be the devil advocate, so we don't have human data yet in this type of preclinical systems. So we are looking for human data, and this is also why the reason our lab is in the ERCnet, all this collaborative consortia. Try to put also introduce in our in silico prediction also possible maybe in 4 years, if you invite me again, I can tell you a much more complete story.
That is the crucial point. So how now translate to this data on humans, and if there is any possibility just to start with the aim of to finish with humans without doing any work.
Yeah. Actually, there is one I was just discussing with you before. So we are lucky because in the US there is really a huge foundation, System Research Foundation, and they want really to move forward drugs from preclinical systems to human for clinical proof of evidence. And we are doing something with Francesco. I'm testing one of the compounds that I named the SGLT2 inhibitor, because we have a quite very nice data set in preclinical systems that seem to reverse proximal tubulopathy, and then we are in the rush to now to move this testing also in human, doing first a pilot studies, and then if successful, apply for clinical trials.
Other questions? I think you and Francesco have some common partners and some common source about this fascinating fact about groups accumulation the cells, because actually we both are just going in the same direction.
Yeah. Actually, well, we had, I think that we were in Heidelberg, together, where you were showing your last findings and we started discussing from that time, with a good beer, German beer. Yeah.
For the young investigators, that of which use it, yeah. Okay. If not,
Maybe.
yeah, sure. Why not?
Just a bit, which was the survival rate?
No, this is a good point. So we actually they don't die. If you take for longer, until 6 months, longer we cannot do because we are not covered by a license, animal license. But until 6 months, I can tell you they don't die. Yeah. So suggesting that probably there are still some. Yeah. So the onset of the proximal tubulopathy is early, much more than compared to the mouse graft. So this is the reason why we are using this system to screen drugs. Because the first point, we want to see how much one drug may reverse proximal tubulopathy before moving, downstream and check other readouts. But no, they don't die. No. Suggest that maybe there are some cross-species difference that may give to zebrafish some sign of longevity. No, unfortunately not. Yeah, we take so from disease, mice with disease phenotypes, in this context, female. Yeah. But then we don't have any problem then if we take sort of rats. So then we don't have any gender difference. Yeah. The beauty of animal species, isn't it?
Okay. Thank you. It's okay. Thank you again for your lesson. Yeah.